Good evening, everybody, and welcome to Current Clinical Trials for Patients with Prostate Cancer with Dr. Gary Ulaner of Hoag Hospital and USC Norris Cancer Center. We are honored and excited to have him back on this Thursday, just before Memorial Day, to discuss clinical trials. Our presenter here is Director of Molecular Imaging and Therapy at Hoag. He is also a full member of the Norris Comprehensive Cancer Center. His funding came from the National Institute of Health as well as the Department of Defense Breast Cancer Research Program. Put that little piece of information in the back of your mind, because he also works with breast cancer research. Dr. Gary Ulaner.
Thank you so much, Neil. I greatly appreciate the opportunity to be here. I'll just say that this will probably run about 20 minutes. I think I sent an email saying we'll go for a target of about 15, 20 minutes. And I'm going to talk about prostate cancer, and in particular, prostate-specific membrane antigen, or PSMA, targeted imaging and therapy, emphasizing the trials that we are currently running or completed at Hoag, and ending with the description of the trials that are currently available or upcoming at Hoag that can benefit patients with prostate cancer. Again, my name is Gary Ulaner, and I am the chair of molecular imaging and therapy at the Hoag Family Cancer Institute, and a professor of radiology and translational genomics at the University of Southern California. And as Neil said, I work on a number of different tumor types, the most common being prostate cancer and breast cancer. But all of my trials emphasize molecular imaging and therapy as what it holds it all together. Here are my disclosures. I also disclose that I like to teach molecular imaging and therapy starting at a very early age. Here's a shot of my son Ilya and my daughter Annabelle helping me teach a molecular imaging and therapy conference a few years ago. And I think I have, indeed I do, a more up-to-date slide of them assisting me at a conference that we now run at Hoag. And as you can see, particularly my daughter has become very adept at finding the important findings on these scans. So I hope that maybe one day she'll be following in my footsteps as someone who's promoting molecular imaging and therapy for patient care. This is the cartoon that I use to describe how molecular imaging and therapy works. My trials are all for patients with cancer. So I say there's a cancer cell, and those cells express different targets. Those targets can act like keyholes, right? Locks on the cancer cell, and we can develop keys which bind to those targets like a key fitting into a lock. The key is composed of a few different components. There's a binding agent that fits into that lock, the key, the ridged port of the key, and then that binding agent is linked to a radioisotope. Radioisotope is something that emits radiation. If the radioisotope emits a small amount of radiation, like we call a positron, then we have built a key that is an imaging agent. It's a molecular imager. It finds the target by binding to it, like the key fitting into a lock, and the radioisotope is emitted. It's something that we'll be able to visualize the products of on what's called a positron emission tomography scanner, or PET scanner, if anyone has had a PET scan. So those are imaging agents. If we take off that radioisotope that emits a low amount of energy and replace it now with something that emits a very high amount of radiation, something like a beta particle or an alpha particle, those are very, very highly energetic particles, Then we have a key that can bind to the target on the cancer cell and emit enough radiation right adjacent to the cancer cell that we can kill cancer cells. So those are molecular therapies. And that's why we call this field molecular imaging and therapy. We build these keys that bind to targets on cancer cells, and then the radioisotopes either emit a small amount of radiation enough for us to image the cancer cells, or a large amount of radiation enough for us to kill the cancer cells. So today, this model, what we'll be talking about is the prostate cancer cell, and the target is a molecule called prostate-specific membrane antigen, or PSMA. PSMA is a protein that sits in the cell membrane of the prostate cancer cell. So here's the membrane of the cell. It's kind of like this. The PSMA is this lock, this target that's sitting on the surface of the prostate cancer cell. And the key are different molecules that bind to the PSMA, like that key fitting into a lock. The one that we'll discuss first is an imaging agent called Polaris, where the radioisotope is fluorine-18, which emits a positron, which is a low energy emission and is able to image prostate cancer in a way that was never done before. All right, so here's our model. And in this model, we got the prostate cancer cell, the PSMA on the surface of the cell, and we're going to start talking about our first key, Polaris, which allows us to bind to that PSMA and image prostate cancer. This is what a normal Polaris scan looks like. If any of the gentlemen who have prostate cancer on the call may have had one of these scans before, this is what you're supposed to see if you don't have any cancer on the scan. You see your lacrimal glands, your salivary glands, the liver, the spleen, the kidneys excrete the agent. That's how it's removed from your bloodstream. And then you excrete through your kidneys to the ureters and the bladder. And so this is a start of what these scans look like. And at Hoag, we ran a clinical trial of this agent in two groups of patients. One group of patients that had what we call high risk prostate cancer that is at high risk for metastasizing at initial diagnosis. And the other group of patients had treated prostate cancer and they had biochemical recurrence, which means their PSA was elevating. Now, these things sound the same, but PSA and PSMA are two entirely different molecules. PSMA is what we're using to target the imaging and therapy of prostate cancer. It's a protein that's in the cell membrane. PSA is something that's secreted into the bloodstream. So I'm sure people who have had prostate cancer have their PSA monitored. That's, again, something secreted into the bloodstream. They take a blood sample, they can look for PSA. PSMA is not found in your bloodstream. It's found on the surface of the prostate cancer cells, and that's what we're imaging or treating with these therapies. Right? So there are two groups of patients that underwent a prospective clinical trial, and I'm not going to go through all the pieces of it, but the bottom line information was among the patients with high-risk prostate cancer at initial diagnosis, this type of PSMA-targeted imaging detected about 1 of 4 patients who had disease that had spread beyond the pelvis and dramatically changed how they would be treated. And in the second group of patients, those that had their rising PSA, about 60% of the patients had lesions that were subsequently biopsy proven to represent sites of prostate recurrence that were not found on other imaging methods like CT or bone scan. So this type of targeted imaging, PSMA targeted imaging, is a tremendous advance over the prior types of imaging that were used to find the sites of prostate cancer in a patient with prostate cancer. We can now more sensitively detect disease, which means patients can be more accurately staged and the proper treatments can be determined for these patients. It's really a new era. And if you have prostate cancer that is high risk, or if you have biochemically recurrent prostate cancer, you're almost assured you're going to be receiving one of these PSMA targeted imaging scans. Here are two examples from that clinical trial to show how this works. This is a patient with prostate cancer post-prostatectomy and radiation, and they had a rising PSA of only 0.6. The CT and bone scan didn't find any evidence of disease, but the Polaris PET scan found an area that was not where we expected. I already went over the stuff we're expect to see. This is an area that we didn't expect to see. And when corresponding it to the CT, we saw a small lymph node, which wouldn't be considered suspicious on the CT because it's too small, but it was very avid on the Polaris PET scan. This white straight structure is a biopsy needle into that node, and it was proven that this was the site of the recurrent malignancy in this patient with biochemical recurrence. This patient could then go on to radiation therapy to the left pelvis, including this lymph node, and their PSA decreased because we effectively targeted. We first found where is the site of the disease, and that allowed us to effectively treat the site of the recurrent disease.
If I could,
Yeah.
Doctor, we can see you clearly. What we can't see is your slides. Is there
You can't
some
see
way
my slides?
I can't?
Something is wrong.
Yeah. Can you enlarge your slides because you're sharing them, but they're small?
Really? Because
Is everybody
they're
else having
full
that problem?
screen. They're full screen on my screen, meaning you
Wow.
can't get any
Okay.
bigger. Let's see
Yeah.
here. Let's try this. I'm going to stop
The-
the share, and I'm going to try sharing it again. And
May
let
I,
me see.
may I interject?
Yeah.
Sure, please.
On the top where it says view options.
There you are.
Click
Is
that
view
better?
options and you can reduce it
Perfect.
to 50% and then it's perfect.
How is this
Yay.
okay?
Yeah.
So let's
Okay.
go back just a little bit. All right. Were you able to see this model where I explained how molecular imaging and
Yes.
therapy works? Perfect. And then here I explained this is our target, the PSMA molecule. All right. And I showed an example of what it looks like normally. And now here's our example of a patient receiving the Polaris scan on the clinical trial. And this is a tiny lymph node that wouldn't be considered suspicious on CT scan, but it's very avid for the Polaris. It gets biopsied. Here's the biopsy needle that proves this is metastatic prostate cancer. And then the patient goes and gets radiation to that node and does very well. So thank you for letting me know that there was a problem in the projection of the slides. This is hopefully much more valuable to people. Here's the second case example that I wanted to discuss, again on the clinical trial. Here's a patient, a 72-year-old gentleman who had a prostatectomy, biochemical recurrence with a PSA of 0.9. The CT and the bone scan were negative, no evidence of disease. Here is the Polaris PET scan showing the normal lacrimal glands, salivary glands, liver, spleen, kidneys, the normal stuff. But unfortunately, there were multiple areas in the chest that were probably not going to be good news. One of them is a very small lymph node metastasis, and another is a focus within a vertebral body, a bone, which here's that white biopsy needle coming in. and going into the site of that was found on the Polaris PET scan, and this was proven to be recurrent prostate cancer. Because this person had multiple sites of disease, it wasn't really very well localized. They weren't a good candidate for radiation therapy to the disease, so their treatment was changed to another systemic therapy in order to cover the multiple sites of disease that were found on the Polaris PET scan. So this imaging modality really has revolutionized the imaging of patients with prostate cancer, both at initial diagnosis and whenever there is a biochemical recurrence. In addition to this imaging, as I said, you take off the fluorine-18 radioisotope that's used for imaging, and you put on something like lutetium-177, sorry for the strange word, but this is something that emits a high energy beta particle. You now have a therapy where you can administer this into a patient's bloodstream. We did several of these just today in my clinic, where it binds to the prostate cancer cell through that PSMA molecule, and it's linked to a high energy emitting radioisotope, and that can kill cancer cells. This is data from a very important clinical trial called Vision, which led to the FDA approval of the very first PSMA targeted therapy, demonstrating that the patients who received this therapy, this PSMA targeted therapy, did better, had longer survival of patients who received other, what they called standard of care therapies. So patients did better with the PSMA targeted therapy than they did with what they were otherwise going to receive without that therapy. These therapies, Hoag has 3 trials that are now closed to accrual, but we still see patients that were accrued onto these trials. So we see many patients for these PSMA targeted therapies. And I wanted to show you a remarkable example of this, of PSMA targeted therapy, on what's called the Eclipse trial. Or this is our patient before PSMA targeted therapy. And if you remember, you're supposed to see those lacrimal glands, the salivary glands, the liver, the spleen, the kidneys, the bladder. But all of this are unfortunately hundreds of sites of disease in the patient's bones. And after 2 cycles of therapy, that disease has dramatically decreased. And this patient had a PSA of greater than 100 prior to therapy, decreased to less than 1 on therapy. So these PSMA targeted imaging and therapy techniques are incredibly powerful, and more patients with prostate cancer will be seeing them. The imaging earlier in the course of disease, and if needed, later in the course of disease when it's metastatic, these PSMA targeted therapies. Okay. So I want to tell you now what's available to patients at the Hoag Molecular Imaging and Therapy Center. 3 of these 4 trials are open. One will likely have open next month in June. The first 3 are the ones that are currently open. 2 of them are imaging trials. The Mirror trial is available to patients with newly diagnosed what's called favorable intermediate prostate cancer, usually patients with Gleason 3+4 prostate cancer. These patients normally don't get imaging of the whole body to evaluate for whether they have a more widespread disease or not. But we have a trial for patients with those Gleason 3+4 newly diagnosed prostate cancer where they can get the Polaris scan. Normally, like right now, patients can't get the scan if they're 3+4. They can get it if they're 4+3, 8, 9, or 10. But if you have colleagues, friends that are newly diagnosed 3+4 Gleason prostate cancer, you may consider letting them know about us at Hoag. We may, if they meet the inclusion criteria for the trial, get them on a trial which allows this PSMA targeted imaging, which could help best define where their disease is and therefore what is the best therapy that they need. That's one trial available for patients with Gleason 3+4 newly diagnosed prostate cancer. The second is for patients with newly diagnosed higher risk prostate cancer. Those are your 4+3s or 8s, 9s, and 10s, right? The 8 is usually means a 4+4. The 9 is either a 4+5 or a 5+4, and the 10 is a 5+5. Of course, the higher those numbers, the more higher risk the prostate cancer is. For those patients, we have a new PSMA targeted imaging agent called copper-64 PSMA. This is not FDA approved, but it is being evaluated to see how well this agent works. And we will get patients with the newly diagnosed 4+3, 8, 9, or 10 Gleason prostate cancer. We'll get them the copper-64 PSMA scan on trial, and then get them the standard Polaris scan so that they get essentially two PSMA targeted imaging studies that we can compare head to head, right? Also, for patients with biochemical recurrence, those are patients who are treated and their PSA is rising. We have the same trial, which we can get both the copper-64 PSMA PET on trial, and then they get the Polaris PET scan as a standard of care, and we're able to compare the two head to head. And in this case, in this trial, essentially labeled number 2, patients can get 2 imaging studies when normally they'll only get 1. All right. Then for PSMA targeted therapy, that agent that I showed, that has such a remarkable therapy potential for patients with metastatic prostate cancer, we have a trial of this medication, which doesn't have a fancy name yet because it isn't FDA approved, but likely shortly will be. It's currently called PNT2002. Now, the PSMA targeted therapy that is FDA approved, called Pluvicto, is only available to patients after they've had androgen receptor therapies like abiraterone or enzalutamide, as well as taxane chemotherapy. And taxane chemotherapy has a lot of side effects. So a lot of patients with metastatic prostate cancer are looking for options other than chemotherapy. We have that clinical trial with PSMA targeted therapy for patients who have disease that's progressing after the androgen receptor therapies like abiraterone or enzalutamide, which are also known as Zytiga and Xtandi, but for patients that haven't had chemotherapy yet. So we can get patients who haven't had chemotherapy yet these a PSMA targeted therapy, when normally, for standard of care, they have to wait till after chemotherapy in order to get the PSMA targeted therapy. And then finally, a trial that we're trying to open up next month is of actinium PSMA, and actinium emits alpha particles, which are 100 times stronger than the beta particles emitted by lutetium-177. So for patients that have metastatic prostate cancer, have received Pluvicto or another lutetium-177 PSMA targeted therapy scan and are now progressing, we can get them on an even stronger PSMA targeted therapy called actinium PSMA. And this medicine has been shown to help patients respond to therapy, even if they're progressing on something like Pluvicto. Now, that's a lot of information in a very short period of time. So I apologize for the density of that information. Let me just review again the things that we have currently to offer patients with prostate cancer in certain clinical scenarios are 2 imaging trials, one of them for patients with newly diagnosed Gleason 3+4 prostate cancer, and the other for patients with newly diagnosed higher Gleason scores from 4+3 to 10. And then for patients who have metastatic prostate cancer that are progressing on androgen receptor therapy, we have a PSMA targeted therapy that we can provide to patients even if they haven't had chemotherapy yet. And then for patients that are progressing after having Pluvicto, we have an even stronger PSMA targeted therapy that can be effective even when Pluvicto is no longer effective. So we have a number of trials at Hoag available for patients with prostate cancer that we hope we can bring a lot of good to this patient population. This is where I'm lecturing from right now. This is my office at the Molecular Imaging and Therapy Clinic here at the Hoag Family Cancer Institute. We have a wing in the Irvine Hospital campus, which is dedicated to these molecular imaging and therapy treatments and trials. We currently have a total of 24 clinical trials open for different types of cancer: prostate cancer, breast cancer, neuroendocrine tumors, myeloma, lung cancer, colon cancer, head and neck cancer, others. But for your prostate cancer imaging group, I really wanted to emphasize what are the opportunities for trials for getting things that you can't get anywhere else, that we have available to patients right here in Orange County at Hoag. If people have questions, this is my email and our clinic phone number. If you would like to reach me, Beth is actually currently out on a leave of absence, so don't email her. Feel free to email me, and I would be happy to answer questions from the group.
Well, Ira has a question, as he often does. In the MIRROR study, can a patient with a small amount of 3+4 qualify for the study? Do you continue to do PSMA on patients who've shown a previous negative PSMA?
The first question I heard was, can you get into the MIRROR trial if you just have a small amount of 3+4? Well, when you do different biopsies, each biopsy gets a Gleason score, right? So maybe you have one biopsy that's 3.4, another biopsy that's 4+3, another biopsy can be 4+4. Your overall Gleason score is determined by your highest Gleason score of a biopsy. So if you have any biopsies that are 4+3 or higher, then you're not eligible for the MIRROR trial, okay? On the other hand, if you have a newly diagnosed prostate cancer that has one of those higher Gleason scores from biopsy, then you're eligible for the copper 64 PSMA trial. You may be eligible for that trial, where we can get you both this PSMA targeted imaging as well as the currently FDA approved Polaris scan. So you can get 2 scans and essentially only have to pay for one. And then we can get the information from both scans to help you with your staging and treatment. So the MIRROR study is only open to patients with Gleason 3+4. And if any of your biopsies are higher numbers than that, then I'm afraid you're not eligible for the MIRROR trial. And then the second question, Neil, can you repeat the second half?
Doctor,
Oh,
could
yeah.
I ask on the first question?
Yes.
A lot of times when some people have a 3+4 with low involvement, say 5%, they could go into an active surveillance program because it's not likely to progress. So that's the 3+4 I'm referring to.
Sure. Well, the patients on the mirror trial are the patients who are being recruited onto the mirror trial. It's for patients who are going to go on to a therapy like prostatectomy, so that you can compare the imaging results to the pathology after the surgery. So if you're a 3+4 that's on surveillance, great. Just stay on surveillance. Neither of these trials are at this point in time for you.
What if I'm a patient with biochemical recurrence after prostatectomy and I'm not doing well with ADTs? Either I don't tolerate them well or it's not stopping reoccurrence. Should I be looking at these studies?
Well, I will there's a algorithm, right, for most patients with metastatic prostate cancer. ADT tends to be the first line. When ADTs stop working on their own, most patients move on to an androgen receptor targeted therapy. As we said, like abiraterone, which is Zytiga, or enzalutamide, which is Xtandi. Right? So that if you're progressing on just ADT, your next step is almost certainly going to be something like Zytiga or Xtandi. If you're progressing on Zytiga or Xtandi, or have or progressed on both, then most doctors start looking at chemotherapies as your next line. That's where, for example, this trial here can be very useful, because we can get you PSMA targeted therapy, which has fewer side effects than chemotherapy, before chemotherapy. Whereas right now, the only FDA approved PSMA targeted therapy is Pluvicto, and the label on that drug is only for patients who have had androgen receptor targeted therapy and chemotherapy. So if you're progressing on just ADT, I think your next line is another standard of care medicine, either Zytiga or Xtandi. If you're progressing on Zytiga or Xtandi, or some of the other androgen receptor agents, there's like darolutamide and apalutamide that are less commonly used. But if you're progressing on one of those, then this trial might be for you.
Very good. I'm going to open this up. JR, do you have a question?
Oh, yeah, actually I do. Can you hear me?
Yes.
I can.
I was just recently placed on, this is to do with hormone therapy. I had the operation 10 years ago, and I've done pretty good, except my PSA went up, and that very imaging that you were talking about, I just had recently, and it was phenomenal. Just, they were able to target it right away. They found a tumor in the bed of the, where the prostate was. So I've been in 3 weeks now of radiation. They were going to put me on Lupatron, the injection, but I asked them if I could take the oral one, which there's only one available now. As you know, I can't pronounce it, but it's spelled ORGOV.
Orgovyx.
Yeah, that one. And so they put me on that last week. What is your take on that? I just had a kind of a bad reaction to it. We're going to take a break and I'll start it again next week. But in your experience, if you think that is a good way to go, it said there were less side effects from that, and that's why.
Well,
and plus,
on average, there are less side effects, but every person is an individual, and you never know how any individual is going to react. So you can try the Orgovyx again, And if you get side effects again that aren't very tolerable, then you may go to the Lupron shot.
Okay.
Right. So you are what I call, still early in the course of disease. You have Orgovix as an option to you. Lupron is an option to you. If those ADTs don't control your disease, then you're going to move to Zytiga or Xtandi. So you as an individual have many options available to you and many options before you would be considering what I have to offer.
Okay. That's good to hear. I know there's an explosion in the treatments. I'm going up to the Leonard Cancer Center now at Mission Hospital, and they've been very good so far, I have to say. But I've always been interested in Hoag because my doctor, Moses Kim, if you know him.
I know him.
Yeah, he was my surgeon. And so, yeah, I just find this so interesting and very helpful. I appreciate your time here and listening to us and giving us this valuable information.
No problem, JR. It is my pleasure.
Thank you.
I'm going to go to Deb. I'm sorry. JR, did I cut you off? Do you have anything else?
That was good. I just, I've got
Okay.
options, so that's great.
Outstanding. I'm going to go to Deb King, and then Bruce has a question he submitted versus chat, and Deb King has her hand up. Deb, welcome back.
Hi, thank you. Thank you so much, Doctor. And I apologize if I missed this because I joined a bit late, so my apologies. But my husband has been diagnosed with stage 4 metastatic disease, and he's been through a variety of treatments. But he's currently scheduled for another PSMA PET scan, and I was wondering what the difference is between the PSMA PET scan that he has with the gallium and all of that versus the copper that you're referencing.
So there are a number of them. First off, there are 3 PSMA targeted PET scans that are currently FDA approved. One is the gallium-68 PSMA 11. One of them is Polarify, the one that I favor. And the third is called Posluma. And they're all very, very similar. If anything, I think the Polarify might be a little better than the other 2 for some more complicated reasons. The physics of fluorine-18 is superior to the physics of gallium-68, and I don't want to go too much into detail. Sure. But you get better resolution images with the fluorine-18. And then there are some biological differences between Polarify and Posluma. So to me, I think the bottom line is all 3 of them do a phenomenal job. And from 0 to 100, they're all way up here to the top of the scale. And if I were going to choose one that may be the best of the 3, I favor Polarify.
Polarify. Okay. And you offer that there at Hoag?
Yes, we do.
Okay. Perfect. Thank you so much for all this information. I really appreciate it.
What about the copper?
The copper one is not FDA approved. It's only being done on clinical trial. So you can't go to your normal physician, your normal radiology shop and ask for a copper PSMA. You have to have it be performed on a clinical trial like one that I have open here at Hoag. We don't know if the copper 64 PSMA is going to be a little better or a little worse than the other ones that are currently available, which is why the way I'm going to run this trial is we'll get you the copper 64 PET scan, PSMA PET scan first on the trial, and then we'll do the regular Polarify, which I think is the best currently FDA approved agent. So at worst, you're getting whatever is currently best. And at best, you're getting something that's even better and is, the next generation.
Excellent. Bruce, you've been waiting patiently. Would you like to unmute and ask your question directly?
Yes, thank you. I don't know if you can hear me or not.
I can.
Perfectly.
Okay, thank you. My question pertains to PSMA. You had mentioned PSMA on the cancer cell itself.
Yes.
Now, is PSMA actually appearing on the membrane of the PSA cells that the prostate cancer makes?
I'm sorry, I didn't understand the question.
Okay.
Is so the prostate cancer cell, right? If my pen is the PSMA, let me see if I get the angle correct. The PSMA is a molecule that sticks from outside the cell, crosses the cell membrane, and extends inside the cell. That is PSMA, and it is expressed on the vast majority of prostate cancer cells.
Okay, so is it expressed on the PSA cells that prostate cancer makes?
Oh, okay, very good. Again, so the prostate cancer cell, all right, we're getting, like I said, there is some difficulty sometimes with the distinction between PSA and PSMA. PSA is made inside the prostate cancer cell,
Right.
and gets secreted into the bloodstream outside the cell, and then is carried around the bloodstream. So when you do a blood test, they would draw a little piece of blood. They can detect the PSA in your blood.
Right.
PSMA is this molecule that's in the membrane of the cancer cell. The vast majority of prostate cancer cells make both PSA and PSMA. There are a small percentage of prostate cancer cells that lack PSMA, but still make PSA. And there are also a very small percentage of prostate cancer tumors that have PSMA, but don't secrete PSA. So the prostate cancer cell in most patients is making both PSA and PSMA. And we can use the PSA in the blood test to help determine if the disease is getting better or worse. And we can use the PSMA on the surface of the cells to image the cells and to treat them with these PSMA targeted therapies.
Thank you. Very helpful differentiation. Thank you.
Bob, would you like to unmute yourself and ask your question? We have a small group here tonight, so.
Yes. In regard to recurrence years after prostatectomy, do you have any numbers on the probability, whether you're using Polaris currently or the copper going forward, as the PSA goes from, let's say, 0.6 to 1.0 or higher? In other words, basically, if nothing shows at 0.6 or 0.7, you have a better, how much better shot do you have at 1.0 or above?
That's a very good question. Excellent question. Because just because your PSA is rising doesn't guarantee that you're going to find the cancer cell on the PSMA scan, right? The higher the PSA number, the more likely you are to find the disease on the PSMA scan, all right? I will start looking for it around 0.2 nanograms per ml. And at that point, you're only going to find that the PSMA scan is only going to be positive in about 30–40% of patients. All right. As you get up to 0.5, 1.0, 2.0, and higher, you keep going. Once you get to 5.0, you're at about a 97% chance that the scan is going to be positive. So there are numbers for all of these, but I don't think it's important to remember all the percentages other than as the PSA goes up, you're more likely to find the disease on the PSMA scan. And if I have a patient with, say, a PSA of 0.6 and the PSMA scan is negative, if the PSA doubles, now say to 1.2, that's about my time point where I'll try using the PSMA scan again, see if we can find the disease that we couldn't find when it was only 0.6. That sound like some good rules of thumb?
Yes, that doubling rule of thumb sounds very good.
Would anybody else like to ask a question?
One more question for Dr. Ulaner. The copper PSMA PET imaging trial, how far in the future is that going to be active
This
for
is
new
well,
recruits?
Well, again, it's active now on clinical trial.
Right, that's what I meant.
It's gonna take probably several years before the clinical trial completes and the data goes to the FDA for FDA approval. But it's available now.
Okay.
On trial.
But going forward, how long do you expect it to still be available to enter?
Oh, on trial? I expect it will probably be available for another year or two.
Okay.
Right? Because the trial will run for a couple of years before I think they need something about like 350 or 400 patients to accrue on the trial to get the data that's needed. So this trial will be open for a while.
Okay. Very good. Something to keep in mind for me.
All right.
Outstanding. Anybody else? Hi, Ira.
I just like to make a comment that, you know, the trials you have are exciting, and especially, you know, the mirror trial, because you're bringing the treatment, when a person's first diagnosed with a small amount. So, you know, it's very exciting. So, so let me just clarify. So you do a PSMA to determine if there's a cancer, cancer outside the prostate, and then it's treated with LU, or is there another treatment? Or what?
So yeah, the mirror trial is for people who are just newly diagnosed. So we do the Polarify scan, and it's not common. Like right now, Polarify is for patients that are here at the stages of Gleason score that you were using on the copper trial, right? So the copper PSMA is where you currently would use Polarify. That's why I'm going to use copper PSMA and Polarify in these patients. The mirror trial is for someone who normally doesn't get any imaging, but you get the Polarify to see if there's anything suspicious outside the prostate, like lymph node metastases. And then the patients go to surgery. And then we get the data from the prostatectomy to see if the imaging was able to find what was found on surgery. The advantage to the patient is that sometimes we find things on the imaging which alters the surgery for the best, or points the patient into a different type of therapy. So imaging before therapy, to me, is why we have this field of molecular imaging and therapy. You image first, and that helps you determine what the best therapies are. So I really love these imaging trials because it helps patients get the therapy that will best help them and give them the best survival and the fewest side effects.
So when they're diagnosed with the cancer, the treatment is to be determined
by the results of the imaging. Correct.
Right. Okay. Well, that sounds, exciting, because people are treating up front instead of later on.
Outstanding. Anybody else have any questions? I do want to point something out tonight, that Dr. Ulaner is part of something very exciting that's happening in Orange County, California. That stands to benefit a lot of people all over the United States and all over the world. And that is Orange County is becoming a major center of cancer research, education, and treatment in its own right. Tonight, we're talking with Hoag, one of the providers down here of outstanding medical care. I've been on the Hoag campus. It is very friendly and very accessible. The Hoag campus, as everything here in Orange County, is accessible through 3 different airports, easy to get in and out of. Ontario Airport in San Bernardino County, Long Beach Airport in Los Angeles County, and of course, John Wayne Orange County Airport, which is very close to Hoag. And we are having some of the top people in the world practicing medicine here, and they're also doing research and education at the same time. If you're viewing this on YouTube, my recommendation is sign up for our newsletters, sign up for our meeting announcements, take a look at our website at www.prostateforum.org. If you sign up for our YouTube channel, you don't get the other benefits you're going to be getting by signing up at prostateforum.org. Take a look at our website. In the upper right-hand corner, you'll see an orange circle with a thumb pointing upward. Click that and join our mailing list or volunteer. We're going to be having meetings for support and information on the 2nd and 4th Tuesdays of every month at 5 p.m. Pacific. We look forward to seeing you there. If there are other questions, we'll entertain questions. If not, thank you, Dr. Ulaner, for joining us. It was a very stimulating session. And as always, we look forward to having you back.
Thank you, Neil. I really appreciate the opportunity to bring, shine a spotlight on important methods of care for patients with prostate cancer.
Outstanding. Thank you again. Look forward to seeing all of you on Tuesday.